So I want to thank Hirofumi Morishita for presenting today. Hiro is, one of our long-term collaborators. He's a associate professor in the Department of Psychiatry, tenured associate professor. He joined as faculty in 2012, and has an MD from Kyushu University and PhD from Osaka University. And he did his psych residency at the National Center Hospital of Neurology and Psychiatry in Tokyo. And he was a postdoc at Boston Children's before he came here. And I think most of us know that his work as a postdoc was really quite revolutionary. It focused on critical periods and whether, critical periods in the development of the brain are plastic or can they be made plastic. And when he began working with vision, which is a classic model for critical periods, and he always had been very committed to trying to make to pivot those critical period studies to psychiatric illnesses. And he's been really kind of an absolute leader in that field. And he's done work on critical periods, and as I said, in vision and perception, but also in cognition and in social behaviors. And he's really kind of created a new model for thinking about critical periods in psychiatry as something that could be manipulated, that is plastic. He's received the One Mind Bipolar Research Award. He's a member of the ACMP. And I'm very excited and pleased, Hiro, that you could give a presentation, and I'm looking forward to your talk. All
right. Thank you, Joseph, for your kind introduction. So like Joseph said, we have been working on a critical period, but today I'd like to share one of the newest line of the research focus on social circuit maturation in the critical period. But before jumping into these topics, I'd like to discuss in a few minutes how the mental perspective is important for understanding psychiatric disorders. So here I'm showing you the burden to the society for many chronic disorders as a function of age of the patient. And what really stand out from here is that when we think about mental disorders, we really need to think about how it's impacting us at this early childhood and adolescent. It's the highest burden to our society around that time. And the reason for this adolescent childhood focus is, of course, their onset and the peak is clustered around this childhood adolescent age across almost any psychiatric disorders. So to really tackle those challenging disorders, we really need to understand what's going on during this childhood and adolescent period. And what is happening during this time is quite complex. It's the combination of maturation of excitatory synapse and inhibitory synapse together with glial maturations like myelination. And we need some conceptual framework to really tackle this challenging moving target. And we have been focusing on the concept of critical period to tackle this challenging question. So what is critical period? We know that our gene interact with environment, but most extensively across development postnatally. And this is the period called critical period when environmental experience can really dramatically rewire our circuitry to help tailor our adult behavior. And we know there's multiple critical periods in our brain, ranging from sensory system, language, and potentially higher cognitive function. And we can imagine disruption of this critical window perceived or experienced during this windows could lead to disorders ranging from amblyopia to potentially mental disorders. So our lab has been interested in dissecting molecular and circuit mechanism governing this critical period phenomenon. And today I will focus on more higher cognitive side of the critical period. I'd like to spend a few minutes discussing how the field evolved using simpler system, which is visual cortical critical period. So critical period for visual cortical plasticity has been extensively studied in the last 50 years, pioneered by Nobel Prize winners Hubel and Wiesel. They find that using kitten and monkeys, early life visual experience can dramatically change the function and structure of primary visual cortex. And this kind of dramatic change is limited only during early critical period, but not in the adult. So this model really set up a stage to dissect mechanism. And one of the key breakthrough for the field was 20 years ago. This was actually the PhD work by our current NIMH director, Joshua Gordon, together with Michael Stryker. They are the first to map the visual critical period in mouse. So by covering one eye for a few days and look at the extent of plasticity by recording from visual cortex, and they find this nice 2-week of plastic window around adolescent age in mice. And this really set a stage, and we made a lot of discovery at the molecular level. I just show you one slide kind of summarizing what the field find. Those in blues are accelerator for enhancing plasticity, and those reds are break for plasticity. And I cited a couple of our contribution here, but we really were able to identify molecular mechanism regulating this visual critical period. So this is exciting, but also open up new questions, which is how this mechanism of critical period find in the visual system could be generalizable to other critical period governing higher cognitive function. I mentioned about this in his book here. So I'm just quoting what he said. Perhaps the most exciting possibility for the future is to extend this type of work to other systems besides sensory. And he said, could it be that deprivation of social contacts early in life may lead to deterioration in some yet unexplored parts of the brain? So this is exactly why when I started my lab in Mount Sinai 9 years ago, we not only continue to study visual system, but also started to expand our study to cognitive and social function, primarily focusing on prefrontal cortex. And the reason we focus on cognitive and social processes is because these are the two key functions commonly disrupted in many psychiatric disorders. And as you know, I think the field is going, instead of studying each disorders, perhaps it's more fruitful to study functions commonly dysregulated in many disorders. And perhaps studying developmental trajectory, we may be able to understand how those processes involved in different disorders. So, and this is what I'd like to focus today. And there's many ways we can tackle this question, but we take kind of a circuit first approach because we think it's kind of the nice middle ground. We know a lot of findings, including Joseph's lab, a lot of genetic risks. That's something we're learning. And on the other hand, you know, a clinical symptom of psychiatric disorder. And I think circuit, how the circuit mature, how risk factor impact those circuit could be a nice middle point that we can bridge all those findings on the molecular side up to symptom level. So our lab has been focusing on trying to identify key vulnerable circuit that mediates key function like social behavior, cognitive function. And today I'll focus on social circuit critical period. So we kind of know how early social experience can impact our brain, especially going through the Corbett shutdown, we can understand the importance of how social isolation, especially early in life, may impact our adult behavior, especially social behavior. And we wanted to understand what are the key circuit that requires early social experience and how that could be damaged by this early isolation. And our hope is to identify vulnerable circuit that will guide us to understand psychiatric disorders. And we wanted to study that in mouse because we can manipulate circuit. And luckily, mouse captures this developmental critical period of social isolation effect. So there is early work where they isolated pups after weaning from P21 for 2 weeks up to P35. This is around the adolescent age for the mouse. And they study the impact of this juvenile social isolation to the adult social behavior. And one aspect of adult social behavior could be characterized by this 3-chamber test developed by Jackie Crowley. Basically, you put mice in the 3-chamber and study how mice will interact with other normal mice versus object. And typically, mouse like to interact with other mice. And early pioneer work by Makinoden et al. Reported that this juvenile social isolation lead to reduced social exploration in the adult using 3-chamber test. So 5 years ago, our former talented PhD student, Lucy Bix, replicated this early finding, but also extended the study by showing there is a sensitive period. So if you isolate mice in the adult and look at their social behavior, we really didn't see this sociability reduction, suggesting there is a sensitive period around this childhood adolescent age that requires juvenile social experience to establish adult behavior. So this helped us to now study the circuit and molecular mechanism of this social critical period. And Lucy reviewed the literature, and we felt prefrontal cortex is a nice place to start. I'm showing you human evidence that multiple prefrontal areas is relevant to social cognition. Of course, mouse brain doesn't capture all aspects of human brain, but at least in part, those areas we colored may have some overlapping anatomical structures. And there's a couple of studies that showed there's a causal role of those brain area for the social behavior. However, we have been missing circuit-specific mechanism governing this key social developmental process. So we set up a question to ask, what could be the key prefrontal circuit that is particularly vulnerable to juvenile social isolation? And we had some clue from recent study. This is a patch recording from prefrontal cortex. And this study looked how juvenile social isolation impact subcortically projecting neuron versus cortically projecting neuron. And interesting finding was that juvenile social isolation preferentially impact subcortically projecting deep layer prefrontal cortex, cortical neurons. So this helped us to set up a question to ask, what is the specific subcortical target PFC to subcortical projection that is particularly vulnerable to juvenile social isolation, and also which is key for sociability. And we are lucky that former postdoc Kazuhiko Yamamuro joined our lab. He is actually a child physiologist, a child psychiatrist, but who can also patch, rare combination of the two, who joined us and tackled this key question: what's the specific prefrontal subcortically projecting neuron that is damaged by isolation and mediate social behavior? So we started from simple experiment to see which area is activated by social exposure. So we used the c-Fos early Mia gene as a marker to look at the whole brain, which area is selectively activated by social exposure over object. And you see here, red lines, c-Fos level after social exposure, black line after object exposure. We look at many prefrontal area. You see frontal area is robustly activated by both stimulus, but perhaps we're looking such a heterogeneous circuit, we don't identify preferential activation to social exposure. However, when we look at subcortical area, we notice one area, which is posterior part of PVT. PVT stands for paraventricular thalamic nucleus. This is located at the top of the thalamus at the midline, next door to habenula. It's a small area, but we see a robust selective activation to social exposure over object. And we felt this could be a nice target of the prefrontal projection relevant to social behavior. And in fact, previous literature suggests PVT is a really nice hub integrating all the neuromodulatory motivation-rated input and sending out also to relevant area like NAP, but also receiving key top-down input from prefrontal cortex. So at this point, we hypothesized prefrontal to PVT projection could be a key circuit for social behavior, and we wondered if it is damaged by social isolation. And PVT, by the way, is also existing in human. It's a thin layer here at the midline, and this also may have some translational impact if we study this. So to test the hypothesis, we like to first check how this particular top-down circuit is activated during the social exploration and how juvenile social isolation impact the activity. So here we put calcium sensor GCAM selectively into this top-down projection neuron and monitor the activity during social exploration using technique called fiber photometry. Basically, we are measuring the activity pattern from this specific projection neuron populational activity. And what I'm showing you is the activity of this top-down neuron here upon exposure to other novel animal versus object in a group house control animal. And you see that this neuron is preferentially activated during the social interaction. What happens in the isolated animal? What we find is that this activation during the social interaction is blunted by juvenile social isolation, as you see here, quite less activation, almost comparable to object exposure, leading to this significant difference in the level of activation to social stimulus. So this result suggests this prefrontal cortex PVT projection requires juvenile social experience to be fully recruited during social interaction. So now the question becomes, is this causally impacting social behavior? So here we now put channel dopsin. This is a special channel that responds to light and change the activity of neuron. Here we use inhibitory type of the channel dopsin, halodopsin. So we are selectively silencing this top-down circuit now during the social behavior in the group house control mice. And what we find is that here you'll see the heat map of the animal to the social side of the chamber versus object. And in a control off condition, you see this preferential interaction with other animal in the control mice. However, when we silence this circuit, we start to see the reduction of the social interaction. We can score that using the sociability score. 1 means all the time interacting with other animal, -1 is only to the object. And you see in off condition, there's a preference to the animal side of the interaction, but this preference disappears after you silence the circuit. You can see preferential social interaction is now masked if you silence this circuit, suggesting that this top-down circuit is necessary for normal sociability in the adult animal. Okay, so what's then happening by the juvenile social isolation in this top-down circuit? What is causing this failed recruitment of this circuit during social interaction? So Kazu patched from the PFC projection neuron using the slice prep, and he looked at how these neurons have how the activation, intrinsic excitability of these neurons, what is the excitatory and inhibitory input to those neurons. Those are the three factors that can change the activation of those neurons. And what he finds is that in an isolated animal, basically the intrinsic excitability was lower. So it's difficult to make this neuron fire. When we look at excitatory drive, inhibitory drive, what really clearly stand out was this huge increase of inhibitory drive. So this neuron is basically on its own difficult to fire, but also it's kind of suppressed by receiving strong inhibitory input. And this pattern was actually relatively selective to this particular projection neuron, because when we patch from other projection neuron, PFC to NAC or cortical projection, we actually didn't see excitability change or excitatory balance ratio, suggesting this particular PFC to PVT projection seems to be particularly vulnerable to the juvenile social isolation. Now, I'm showing you the adult result, and you may wonder how this change happened across development. So Kazuya also patched from developing animal, and this is where we actually find some surprise. So here I'm showing you the intrinsic excitability, how much neuron fire. And the big surprise for us was when we patch from P35, when is the end of the juvenile social isolation. Actually, we didn't detect the difference in the intrinsic excitability. And it was really after isolated animals group house, that's when those deviation from the normal trajectory happens. This was the same when we look at inhibitory drive. So the inhibitory neuron, this black in a group house control, has an interesting pattern. After P35 toward the adult, their inhibitory drive actually decreased significantly. And this normal developmental pattern was not happening in isolated animal, as you see here. So high inhibitory drive at this adolescent age remain high in the adult. So this combination of the change lead to the huge difference in the EI balance in the isolated animal. You see this increase in the normal development, which is kind of stuck at the juvenile level in the isolated animal. So this suggests there's a complex change happening, maybe the mismatch between the isolated environment and the following subsequent group house environment. This mismatch may be non-linearly leading to this deficit, which we hope to study in our lab in coming years. But I'd like to come back to the to further discuss where this hyperinhibition is coming from. So Kazu patched from various GABAergic interneuron, which is quite diverse, trying to find the cell that has higher excitability. And it has been a process. For example, parvalbumin cell relevant for psychiatric disorder, we didn't see activation, we rarely see lower excitability, so as some part of somatostatin interneuron. And we are left with one cell type, which is low threshold spiking type somatostatin interneuron, which are located in a deep layer. And we're lucky to happen to have a Cree mouse line that selectively labeled this low threshold somatostatin interneuron. And when we patch from those neurons, what we find is this elevated intrinsic excitability. And this was something we were looking for, which can fit to this hyperinhibition onto this top-down projection to PVT. And in fact, when we look at the connectivity between the somatostatin neuron and the PFC top-down neuron using optogenetics, basically we stimulated this somatostatin neuron selectively, and when we patch from this top-down neuron, we see a strong monosynaptic connection. But also, when we analyze paired pulse ratio, which look at the release from this GABAergic neuron, we see aberrant pattern indicating more release. So this inhibitory neurons deficit kind of converge to this key top-down neuron. And in fact, if you mimic this hyperexcitability of the somatostatin neuron by putting excitatory dread, this is a special channel that will activate this neuron by giving some specialized drug called CNO. And if you do that in a group house control animal, we actually see this deficit in the sociability kind of phenocopying this juvenile social isolation phenotype, suggesting that this key somatostatin neuron highly connected with this top-down neuron. It is likely part of the deficit causing the isolated animal to lead to sociability deficit. Okay, so now can we rescue the sociability deficit? So here we brought isolated animal, which has sociability deficit, and put excitatory channel opsin to stimulate this circuit during the social interaction. And when we did that, what we saw is the acute online rescue of the social behavior. So here, where is no light stimulation, but when we stimulate this top-down circuit, we now start to see the rescue of this sociability. Here you see how we do. We use this wireless optogenetics to preserve more natural social behavior. And this suggests that stimulating this circuit could be a good therapeutic target for rescuing social behavior. However, when we think about intervention, we want the effect to sustain. So what we did is we repeated this paired stimulation for 2 days. And on the 4th day, we see even without stimulation, can we maintain the sociability rescue? And that's exactly what we observed. So here I'm showing the sociability score from before the stimulation and during the stimulation over 2 days, you see this nice rescue of sociability score, which importantly sustained it in the 4th day when we are not stimulating the circuit. So this gives us a hope. This circuit could be a good target for rescuing sociability, at least in mouse. So summarizing what I showed you, we started asking what are the key circuit that is damaged by juvenile social isolation. And we find that this prefrontal deep layer pyramidal neuron projecting to PVT is recruited by social interaction and is necessary for sociability in the adult animal. However, in the isolated animal, juvenile social isolation, we find that this key social circuit is less excitable, and on the top of that, receiving stronger inhibition from this deep layer somatostatin interneuron. And this combination of this two in excitatory inhibitory neuron pair contribute to failed recruitment of the circuit during social behavior, and also causally leading to the sociability deficit. And at the end, I showed you that we can actually stimulate this circuit to rescue this sociability deficit. So putting all together, we think that there is a particular pair of excitatory inhibitory circuit in the prefrontal cortex that requires juvenile social isolation. And it is actually vulnerable to this isolation effect. But vulnerability is kind of the two sides of the coin. We showed that actually stimulating this circuit rescued the behavior. So we think this vulnerable circuit could be a good target for therapeutics. Now, we are excited we find some key vulnerable circuit causally important for social behavior, but the question is this more relevant to bigger context of disorder? And specifically, does rare de novo variation related to autism, are they also impacting those circuits? So we felt this question makes sense to ask because recent study by Matthew State showed that autistic risk gene, they tend to be enriched in terms of their expression in deep layer pyramidal neurons in the cortex, particularly prefrontal area. And as I said, the circuit we focused on, PFC PVT circuit, they are clustered in layer 6 and deep layer of layer 5. So at this point, we hypothesize maybe not only the juvenile social experience, but perhaps autistic risk gene may impact the early development of this key top-down social circuit, and that may prevent this normal experience-dependent maturational process. So our new postdoc, Dr. Kazuya Okamura, who is also a psychiatrist, has been studying this circuit using mouse model with monogenetic form of autistic risk gene. And through the support by Simon's Foundation, we are actually excited to find really interesting convergence of the autistic risk gene to this circuit. This is preliminary data, but when Kazuya patched from this PFC to PVT neuron, he find that Fragile X knockout or TSC mutant mice, these are the two rare monogenetic form of autistic risk gene, they show this reduced excitability, very similar to juvenile social isolated animal. And when you look at the excitatory inhibitory drive, again, we see something striking, which is strong inhibition, which lead to the skewed EI ratio, which all looks very similar to juvenile socially isolated. So we are now thinking maybe this key circuit, social circuit, could be the converging vulnerable circuit, not only from the developmental and experience dependent part, but also from the genetic risk of the disorder. So we're characterizing multiple autistic mouse models to see if we can generalize this rescue strategies. And also, we're lucky to receive funding from One Mind to study bipolar disorders. So there's an interesting study that showing PVT could be preferentially damaged by some of the bipolar risk gene. So we like to extend the study to bipolar disorder as well. Okay, so I told you about our work in the social behavior and identification of specific excitatory inventory cells that mediate social behavior. I didn't have time today, but I'd like to also share with you what we find in the other prefrontal circuit in the context of cognitive control in coming few minutes. So I told you that isolation preferentially impacts subcortically projecting neuron, but in our study using translational touchscreen behavior, especially attention behavior, our recent work identified that prefrontal projection back to the visual cortex, so-called top-down feedback projection, they play a very causal important role in attentional behavior. But we also find this circuit undergoes key change in the balance of local and long-range input following adolescence. And we find that neuromodulatory system plays a key role in this. And this study also indicates the concept of local long-range balance could be as equally important as this excitatory inhibitory balance concept. So I'd like to share with you a couple of take-home slides from this new line of research. So our former MD PhD student, Elisa Nabel, together with our postdoc, Yuri Garkin, wanted to understand how this PFC to V1 feedback projection mature. And one of the key findings was the extent of local input drastically changed following adolescence. But the long-range input to this key circuit didn't change. So there was this interesting shift of local long-range balance during this key developmental window, shifting from hyperlocal to hyperlocal. And we wanted to understand what is driving this key change. And long story short, we find that during this process, nicotinic signaling drastically dampened following adolescence. And we find that this was mediated by the increased expression of nicotinic brake called LINX1, which causally prune away this local excitatory input. And this was interesting because LINX1 was not foreign to us. It was actually the molecule we find in the visual critical period system. It actually closed this visual critical period. And if you remove this molecule, you have this open-ended, immature form of the plasticity. So in the prefrontal cortex, it is also playing a key role, pruning this local input, and seems like a key driver for this developmental change. So we also looked disease, if there's any model that is compromised with this process. And through the unbiased informatics approach, we happen to find Fragile X knockout model, which is the leading genetic cause of the autism. This model has hyperlocal connection, hyper nicotinic tone with attentional deficit, almost looks like juvenile animal, kind of circuit maturation stuck at the juvenile level with too much local input. Interestingly, we are able to prevent this deficit by dampening down the nicotinic tone by overexpressing LINX1, which end up pruning away this local input and also rescuing attentional behavior. So we think studying this key developmental process will be useful and provide new idea for intervention. I showed you our finding in the social behavior side. There's a specific pair of excitatory interneuron important for social behavior and require juvenile social isolation. I showed you those circuits could be also disrupted by genetic risk of autism. And briefly also shared with you some of our finding in the cognitive side, which indicate not only EI balance, but within the excitatory circuit, maybe we need to think about the balance of local long-range input. And I showed you the relevance in the context of autism model. Okay, so now I'd like to wrap up. So, okay, so this is all promising, but is it really relevant to human disorders? And I'd like to finish by sharing our effort of trying to make our preclinical research in mouse to be translatable to human disorders. And I'd like to share with you what the ongoing brain initiative effort at NIH is heading at. So they recently organized this work group trying to determine what's the high priority area in coming 10 years. And they identified 5 transformative projects for the field. And one of them was reaching circuit cures. So they identified that to achieve circuit level understanding of and intervention for vulnerable circuit as a move forward protecting or correcting a major human psychiatric symptom. So basically the idea is it will be important to identify specific circuit that is vulnerable, and those vulnerable circuit could be the good target for therapeutics. It could be either neuromodulation or it could be drug intervention targeting those circuits. And I think this is a key, this is one of many, but important future goal for us to identify specific vulnerable circuit, but also try to see how that could be relatable to human disorder condition. I'd like to finish by sharing how we aim to reach to this goal. So I'll use the example of the PFC PVT circuit in the context of social behavior. We have been working on establishing a causal link between specific circuit and key behavior like sociability, which is frequently disrupted in disorders. And using chemogenetics and optogenetics, we were able to establish the causal link. But now, what is left for us to bridge to translation is, we think there's two key readout we should focus on. One is to identify circuit-associated functional biomarker. So what I mean is that, is there any electrophysiological change that could be even studied in human, but is more tightly associated with specific function, circuit. So for this, our ongoing work is trying to examine the local field potential change while we manipulate specific circuit. So we could either put child dopsing or dread to silence the circuit while monitoring the local field potential during social behavior. This will help us to identify, say, theta frequency coupling between PFC and PVT, or beta frequency power, for example. That we like to identify brain change, electrophysiological change associated with specific circuit in a causal way. And that will help us when we perform, say, social tasks in human, stratify specific patient group that may have a deficit in certain circuit. But biomarker is not sufficient. We like to find therapeutics. So our second goal to make our research translatable is to identify targets that is enriched in our circuit of interest. And for that, we are fact sorting the nuclei from this specific top-down circuit. And we are interested in finding specific drug targets enriched in this specific circuit, which may be changing during development or changing in the disease model, which will help us to come up with two compounds that may preferentially target those circuits. And once we have functional biomarker and drugs that is preferentially targeting the key circuit that is causally related to key function, we are in a better place for circuit cure. We can test this idea in a preclinical mouse model, but ultimately our hope is having those two readouts will help us to establish parallel human tasks so that we can stratify patient group more from the circuit point of view for effective therapeutics. And I'd like to finish by mentioning one new collaboration in this translational domain. We are lucky to receive a Freeman Brain Institute Pilot Award for our new collaboration with Rita Goldstein and Mount Sinai. She's one of the leader of the human imaging. And we are going to study the same PFC to PVT circuit using similar approach, which is functional MRI. By using same modality of the measurement, our hope is to link some of the change we are finding in mouse. We could put dread in the circuit and see what's network changes and how that related to human functional brain imaging study. So we are excited that to see how much we can translate our finding in mouse to the human disorders. With that, I'd like to finish by thanking our lab member. It has been all the team effort put it to come to this point. And also, we are so lucky to have many collaborators at Sinai, and also grateful for the funding. And finally, I'd like to thank you for your attention to work, and happy to take questions.
Well, thank you, Vera, for a great talk. I see people remember to use the Q&A ideally for questions. If you use the chat, I can also look there, but Q&A is the simple one.
So I'm seeing two questions. Maybe I read and try
Sure.
to answer it.
Yeah.
So first question from Ken Buyukasik. So let's see. So we're interested in rescue approach. What is it about the critical period that allows experience-dependent recruitment of this circuit? Would it be possible to return the mental critical period selectively or more generally rather than experimental circuit activation? Right. So great question. We in our study, we try to identify a few circuits that is vulnerable to this social isolation effect, which has a critical period. I should probably stress our approach was not meant to reactivate the critical period window. I think that's the key question we should study. For example, is links-1 involved in this critical window? If that's the case, maybe we want to manipulate that. So the question of what is it about critical period for the social experience-dependent plasticity, I think we still don't have the answer. And if you remember, I showed you some complex aspect about this critical period. When we examined the circuit right after the isolation, we actually didn't see apparent change. So it's more nonlinear complex process that requires this imbalance even after this critical period, which is different from the visual critical period. When we cover one eye, we already see a change after 4 days of plasticity. So we have a couple of lab members who is trying to understand what's the developmental change even after the isolation from following adolescent. And maybe we find another critical period for intervention that time. Okay, so Hala has a question. Can you please explain how you think stimulation of the PFC to PVT circuit can have a long-lasting effect, even after the stimulation, right? So we don't yet know the mechanism. We suspect there may be some plastic change happening between the PFC to PVT terminal connection to the PVT neuron. So our student, Mike Leventhal, is actually trying to look at how the local field coherence change between these two regions after this optogenetic stimulation. Maybe we could also patch from this and look at the mechanism. So that's a future question we need to address.
I'll have a follow-on question.
Yeah. How can you please explain the manifestation of the ephes deficit core in the adult, but not immediately after isolation? Yeah. So I kind of touch on this, but we think it's a combination of isolation during early period and very different living environment, which is group house after isolation. So this is almost like an extreme mismatch of the experience. And you can imagine animals start to adapt to this isolated living condition during the plastic critical period. And then if you imagine if this window closed, which is still hypothesis, and if you suddenly meet with a bunch of animal, but if critical period is closed, you may not be able to adjust anymore. And this mismatch may start to harm the circuit. So that's one hypothesis we have, and something we like to follow up. But this was one of the big surprise for us, but kind of excited we find some unexpected finding, which we like to follow up.
Thank you. Maybe going back to the question of the timing of rescue. So I know I read it in the paper, but I honestly don't remember. So when you do the rescue, the animals are first exposed, then you treat, like in the first paradigm, then you treat and then you put them back together, or do you treat during the first?
Right. So thanks for asking the clarification. Our manipulation was all in the adult end. So animal went through already isolation and the 1 month of group house.
Yeah.
And then in the adult, we attempt the rescue.
Right.
So,
but the rescue is, but I presume the experiment is you put the mice, you put them, the mouse with the old mouse, mouse A, and then with mouse, A and B, right? And somewhere in between, do the rescue before you put them with the first mouse, or only when you put them?
oh, I see. So we basically simulate a circuit during the social interaction.
The first
task.
interaction.
Right. And when we see the sustained effect, that was also the case. We only stimulated when animals start to interact with the other animals. So we put the area that will stimulate. So kind of pairing the stimulation and social interaction.
So basically somehow reinforcing the social interaction.
Right. So then the question becomes, what happens if you stimulate, say, in a cage, not in the context of social interaction task, and do you even see the effect? Or we could even think about stimulating right after group house. It's clear something is happening between P35 and the adult. Yep. And if you stimulate during this window, can we even prevent the deficit?
Right.
So we have some clue. It could be the case. Our former PhD student Lucy Bix asked this question in the parvamine interneuron. And she actually finds, this is unpublished, we're about to submit. If you stimulate during P35 to P45, you may be actually able to prevent the deficit. So we think window for vulnerability and intervention could be not necessarily overlapping.
Right. So those slopes of the excitatory and inhibitory that sort of starts splitting right after P35,
right,
it no longer happens.
Right. I think that's a key therapeutic window.
Fascinating. Well, I think that was I know that was a wonderful talk. I don't know if looks like people, everybody enjoyed your talk, and I see there are no more questions. So thank you, and I'll also just thank you again.
Yeah, thank you all for joining.
It's great talk. I think a really kind of a fascinating new direction, and we look forward to hearing more and even collaborating.
Yeah, definitely. Looking forward to collaboration with CBER Center.
Thank you.
All right, thank you.
Thank you, everybody, for joining. Bye.